Okay, why don't we get started? Well, what I wanted to do today was start talking about episodic memory and the hippocampus in a more clinical domain. Really, up till now, we've talked about it in terms of healthy, normal humans. The brains you were looking at when you were doing the lab were all from healthy young subjects. And today I wanted to talk about its relevance of studying memory in the hippocampus to disease processes, namely Alzheimer's disease. So just to give you a little overview of what I wanted to talk about today, talk about some of the cognitive changes associated with Alzheimer's disease, and then talk about the epidemiology, so some of the risk factors for Alzheimer's disease that we understand now, and this concept of cognitive reserve, which is kind of what we'll be testing with the behavioral data that you collected this week. And talk a little bit about some of the highlights of the Golda article with some of these treatments on the horizon. And then just briefly talk a little bit about the importance of being able to identify preclinical Alzheimer's disease, which is another very big, important area in this field that... So I'm going to really be talking about very cutting edge, kind of the current view of the field in this lecture today. And as you can tell, the Golda article hasn't even come out in print yet. It's so brand new. It's actually still a galley proof. So it's really the current understanding in the field. Okay, first though, I want to give you a little preview of what we'll be doing in lab 10th week. So you hopefully know that there is no lab or lectures next week, so you'll have a happy Thanksgiving. Obviously don't show up here Friday waiting for a lecture. We're not going to have a lecture. But we will reconvene 10th week in lab. And some of the things we're going to do, we're going to score the data from that Remember No task that you took this week. And then we are going to analyze those data using t-tests to see whether memory for incidental details is more accurate for items where subjects gave R responses versus K responses. And I think you probably have a sense of how that should turn out based on the previous lectures. But we will see if we can find support for that idea. Does anyone have a feeling which way it should go or what way you predict it would go based on my lectures? What should be the memory for incidental details should be more accurate for either for R or K responses? What do you think? R, yeah. So we'll see if there is support for that. That's sort of my prediction of what will happen. And then another analysis we're going to do. So we're going to use t-tests sort of like how you've been doing. But we're also going to do a correlation analysis between the number of relatives that you guys had with dementia and episodic memory performance to see if there's any relationship there. So we'll also use Excel to run a correlation analysis. And then the third thing we're going to do, or actually the fourth thing we're going to do, is we're going to go back to that hippocampal data set that you've been working with these last couple of weeks and look at possible interactions between factors. So this week you went into a lot of detail with group comparisons. So within subjects, looking at psi differences and looking at group differences between sex and age. And it looked like, I think everybody found, in all the sections found a difference within subjects in extent of the hippocampus, with the left hippocampus longer. And some of you also found that the left hippocampus length differed by age. And I think in Thursday that wasn't significant. The other two sections it was. So, but some questions arose, like saying, oh, okay, well, the left hippocampus seems longer overall, but given it seems like at least numerically it's longer in the older subjects, maybe there's a difference in laterality, that maybe the pattern is different between the young and old subjects. Maybe the younger subjects, the left and right hippocampus have a similar length, and it's really the older subjects that are driving that difference. In the older subjects, you see a big difference between the left and the right hippocampus in length. And really, t-tests alone won't be able to tell you what that difference is. We'll need to use what's called an analysis of variance and look for interactions between factors. So particularly like interaction between age and side. And that might show that there's a difference in pattern of the laterality between old and young subjects. So we'll explore that. Also see if there's any possible interactions with sex and size. So it could be that even though overall no one really found any sex differences, it could be that the pattern of laterality differs, that in men there's maybe the right is slightly bigger than the left, and women it's flipped. We can't really tell that with t-tests very well, so we go to analysis of variance. So that's what's kind of on tap for 10th week. Okay, so let me start talking about Alzheimer's disease, most common neurodegenerative disorder that exists, and the risk increases sharply with age. Now here's an example of an Excel-type graph. I got this from a government website and it illustrates one principle that Dr. Grisham said you should never do, is assume that your audience cannot read graphs and actually put the means up here. I think he said that in all the sections, that you really shouldn't do that. But these guys did that. But anyway, what you can see, if you can read this graph, is that as age increases, the incidence of Alzheimer's disease increases sharply. So this is 65 to 74, and it's about 3% of people have a diagnosis of Alzheimer's disease in that age range. Go up to 75 to 84, it's up to 19. And over 85, it's up to 47%. Now, with the aging of the population, we're getting better at fighting things that killed us off earlier, like infectious diseases and cancer and treatments. We have treatments for those things. People are living longer, which is great. However, looking at these numbers, where if people get up into their 80s, 90s, and beyond, the incidence of Alzheimer's disease is very high. So this is actually an increasing problem that's accompanying the general aging of the population. Okay. So what are the behavioral findings? How do we diagnose Alzheimer's disease? What is it? Well, it is a dementia. And what is dementia? The definition of dementia is a memory impairment plus at least one other deficit. So we talked a lot about amnesic patients who have a really severe memory impairment. However, they would not be called having a dementia. So HM, though his memory impairment was exceedingly profound, he would not be demented. Patients with Alzheimer's disease, they won't really get a diagnosis of Alzheimer's disease unless, in addition to having some problems with memory, they have problems with something else as well. Memory might be their biggest problem. But they also have some other deficit, and that is usually a visuospatial deficit. So these patients may have trouble like constructing things, or they might kind of get lost in familiar environments. They may have language problems. They may stop being able to understand language very well, or know the names of things, or know concepts, or reasoning. They might start to make poor decisions. That would be another example. And another important definition of Alzheimer's disease is it's progressive. So if a person, say, suddenly develops some cognitive symptoms and then they sort of stay at that plateau, it probably is not Alzheimer's disease. It may have been because a stroke or some other kind of problem. Maybe in elderly you often have problems with too much medication that can have cognitive implications. So you wouldn't jump to a conclusion of Alzheimer's disease if the person seems to have had a problem and then just sort of stayed stable. Really, another key aspect is it gets worse and worse and worse from year to year. It's progressive disease. That's part of the definition. So if you have these things, diagnosis of Alzheimer's disease is quite likely. Okay, so just some examples, especially of the visuospatial problems that these patients often manifest pretty early. And this clock drawing test is actually pretty effective. It's simple, that a physician can just do in the examining room, and it's actually pretty sensitive to cognitive deficits. So you see a patient who just maybe has early Alzheimer's disease where you're not really sure if they even have it or not, and they can make a clock. It doesn't maybe look great. 1, 2, 3, 4, 5, and you see the 6 is over here, so still, and the 10 is here, and then they remember, oh, 12 has to be on top, 11, 12. So, it's not a great example. It's sort of passable, but you'd think that they should be able to do better. So there's some problems. However, as the Alzheimer's disease progresses, you can see huge problems with visuospatial abilities, in that they're not even able to put the numbers within the circle. They, go 1, 2, 3, 4, 5, and then they kind of know 6 should be at the bottom, but then they go off this way. A big visuospatial problem. And as the Alzheimer's disease gets more progressive, not only do they seem to have problems placing the numbers, but just their order is off here. They have 11, 2, 3, 4, 5, 6. It's just really not placed anywhere near correctly. And it seems like they're actually kind of losing the concept of what a clock is, because the numbers are sort of out of order. So you can see as the disease progresses, even with this really simple test, just draw a clock for me, you can actually see the deficits in these patients. I think in addition, the instructions, draw a clock for me and put the hands at 2 o'clock. Show me that it's 2 o'clock. And they kind of know the 2, but they're still sort of, even at this early stage, have some problem there. So this isn't a memory problem. It's more of a visuospatial problem and an understanding problem. As the disease progresses, they don't even attempt to know how to show 2 o'clock on this clock. Okay. So those are the cognitive findings. What about the neuropathological findings? In fact, to officially get a diagnosis of Alzheimer's disease, you need to have these specific neuropathological changes. In real life, though, people who start to have some cognitive decline, some elderly people, you don't usually go in for a brain biopsy to note these things. You basically make the diagnosis of probable Alzheimer's disease just based on behavior. But however, the real definition of Alzheimer's disease depends on having these neuropathological findings. So its definition is defined by the presence of 2 things: beta amyloid plaques and neurofibrillary tangles. Okay, so these things are present in the brains of Alzheimer's disease. That's sort of the defining characteristic. So both of these features just kind of are basically, if you had to say what are they, it's caused by proteins that are normally soluble becoming these polymers that are insoluble, that they're kind of, these plaques are basically the beta amyloid that gets kind of bunched up in kind of a little glob and in the brain, and the same with the neurofibrillary tangles. These are proteins that normally are soluble, but they become these little clumps. So that's the characteristic pathology in Alzheimer's disease. It's also accompanied by the loss of neurons. Just having a bunch of this gunk in your brain may not necessarily be a bad thing, but it does seem like it is associated with a loss of neurons. Okay. And that's, of course, the problem and why the cognitive deficits appear. Yeah, question? They're separate things. So there's the amyloid plaques, and then you also see the tangles. And you're right. A question is, does one cause the other? Is there some third factor causing both? How are they related? Which is the one that's killing the neurons? Are they both killing the neurons? Or is some third factor killing the neurons plus depositing these plaques? Those are key questions if we want a treatment. And some progress is being made, and I'll kind of describe some of that and what we know at this point. Okay, so the location of Alzheimer's pathology. As I said, Alzheimer's is progressive, and the pattern is fairly variable among patients, but typically you see it start in the medial temporal lobe. So you should be very familiar with sections like this. You spent a few hours staring at them, and you might notice hippocampus here. This is a normal brain, age matched for this person down here, and you can clearly see the hippocampus. If you had to circle it, you could do that. Now look at this person down here, this black hole. This is a more advanced AD case. It's black, and remember that was fluid. They basically don't have anything there. So if you're trying to draw the hippocampus in this advanced AD patient, I don't know what you would actually draw. It doesn't look like there's much there because it's just really fluid. However, it's not like there's a selective problem there. Certainly you can note that it looks like there's degeneration in the cortex. So like if you look at this person, here's the temporal lobe here, and you see the white matter covered by all this gray matter. That would be normal. Here you see a lot of that gray matter degenerated. You really don't see it. Look at these ventricles, how large those are compared to a healthy subject. So you have overall degeneration as well. I think also you just see less cortex here up in the, in parietal regions. You just see a lot of general degeneration. However, some of the earliest regions where you see Alzheimer's disease is in the medial temporal lobe, hippocampus and entorhinal cortex. You start to see in the earliest stages shrinkage and the development of these plaques and tangles. So that's the typical progression. Starts in hippocampus and entorhinal cortex, but then other areas become involved as the disease progresses. And I would say the regions that you, in an advanced case where you see degeneration, temporal, parietal, and frontal lobes particularly. And that kind of fits. Parietal, would be involved in visuospatial, which is a symptom that comes on pretty early. In these patients, even in fairly advanced stages, you see sensory cortices and motor cortex can be fairly well preserved, I think, relative to the other cortical areas. So it seems these high-level association cortices that we talked about, which are, of course, the regions that in a previous lecture we talked about are the regions that are really feeding the medial temporal lobe information. Those are areas that also seem to be degenerating in Alzheimer's disease as it progresses. Okay, so little highlight of what these plaques and tangles look like. Here's some plaques. Here's another one, this brown thing. Here's some little tangles. So what are the plaques? Well, you have a protein in your brain or in your body, amyloid precursor protein, and it can be broken down into fragments that are called beta amyloid fragments. Normally, you would be able to process these and break them down, these beta amyloid fragments, but in Alzheimer's disease, they seem to form these clumps. So these clumps are clumps of beta amyloid that are not broken down, that are just kind of sit there as big clumps. What about tangles? Tangles are formed from the tau protein. So you may have heard of this protein in terms of it's a component of microtubules. About microtubules, probably have heard about those. Of course, they're very important for the structure of the neuron. But, so it's an important protein, but in AD it looks like it forms these tangles that are hyperphosphorylated. There's a lot of they get all phosphorylated and they form these kind of pathological little tangly clumps that a normal brain doesn't have. Okay, so that's kind of the pathological signs that define Alzheimer's disease. They have these two different things. Now, I should say that there are a lot of people, healthy, elderly, running around that have, that have maybe donated their body and brain to science, and even though they were very high functioning, no one had diagnosed them with AD at all or dementia. You can notice some of this pathology in their brain. So there are healthy people with this, you can find plaques and tangles. Okay. So, well, how is it caused? We don't know for sure, but certainly there are a lot of good theories out there. I'd say the dominant view, and certainly the view in the Golda article, focuses on the amyloid plaques. And the idea is that because there's a problem with processing the amyloid precursor protein and breaking it down into these amyloid fragments, that they clump, and the amyloid plaques are directly neurotoxic. Remember, the problem with Alzheimer's disease is not that you have this gunk in your brain, it's the cell death. That's what's causing the cognitive problems. And that these plaques appear to be directly neurotoxic. What's the evidence for that? Well, transgenic mice, which overexpress the amyloid precursor protein, and as you probably know, we have techniques now where we can create strains of mice where we can manipulate the genetics of the mouse, and we know the gene that codes amyloid precursor protein. So when we create these mice that overexpress this protein, they do have a lot of plaques. They also have a loss of neurons. Okay, so that's one big piece of evidence, a direct piece of evidence that actually that plaques are killing the cells. Also, there are people who have genetic problems leading to overexpression of APP, or the amyloid precursor protein, that because sort of like these transgenic mice, they have a mutation and that causes an overexpression of APP or a problem with processing it. And this genetic mutation leads to a very early onset, so like people in their 50s and their 60s in these families get Alzheimer's disease with lots of plaques, suggesting that, hey, even if you have a real problem with your amyloid precursor protein or processing it, that's really a cause of AD. Let's work on fixing that to come up with a treatment. Now, most of the people running around with Alzheimer's disease don't have a mutation in the APP protein or any kind of gene that codes for these things, but the small subset of Alzheimer's disease patients that have been well studied to try to make that link between the toxicity of the amyloid plaques and cell death and the disease process. Okay, now there are some alternative hypotheses out there, and they're briefly touched on in the Golda article. One problem or one reason that people have been looking at alternative causes is there isn't a super strong correlation between plaques and cell loss. You can, like I said, you can find people who have a fair number of plaques and they don't have a lot of cell loss. So it doesn't seem like it's a really super tight correlation as much as you might expect if that's really the cause of AD. And then looking at the tangles, well, other neurodegenerative diseases that are rarer than Alzheimer's disease, but things like frontotemporal dementia, those are definitely caused by problems with the tau protein. So in, say, frontotemporal dementia, you don't have plaques, but you have real problems with these tangles and the tau. And that causes dementia and progressive cell death. And of course, when the tau isn't functioning properly, it's an important component of microtubules, you can see how that could kill cells. If the microtubule protein is not really functioning properly, that's causing cell death. It's less clear how the beta amyloid plaques are really killing cells. There's some theories out there, but it's less obviously clear. And of course, maybe there's a third factor supporting both, that there's some other disease process. One consequence is it causes plaques, it causes tangles, but this other thing is really what's killing the cells. And so if you supported this view, you might think, well, gee, maybe you shouldn't put all your eggs in one basket trying to treat the beta amyloid when there's actually some third process that's actually killing the cells, and just dissolving the plaques isn't going to really cure the Alzheimer's disease. I would say that this is somewhat of a minority opinion, that really given the support for the amyloid hypothesis, most of the research has been directed towards that. However, there is some other research going on looking at tau, looking at some of these other factors. So just be aware of that. And I think the Golda article did kind of lay that out, that both of those things are going on, although the amyloid hypothesis seems to be the leading contender at this point. Okay, so what about risk factors for AD? Maybe that can also give us some idea about how to treat it, but also maybe prevent it, which of course would be what we'd really like to do. As I said, there's some genetic risk factors. First of all, there are some specific mutations that have been reported that are directly linked to an early onset AD, and they tend to be autosomal dominant inheritance. And so a few different pedigrees have been described where the family has, a lot of people in the family get an Alzheimer's disease, say, in their 50s, and it has a pretty bad course. I mean, they get worse pretty quickly. And they've been studied in terms, and usually like half the offspring of a parent who has this genetic disorder will also get it. So there's several families that have been studied in a lot of detail. And in some of these pedigrees, they have detected the gene seems to be either on, there's some on chromosome 21, 14, and chromosome 1. There's some families in Japan and one in Sweden, several in the United States that have been studied really extensively. Now, the majority of patients with Alzheimer's disease don't fall into this category. However, these genetic cases have been very interesting because we can see what is the gene doing that's mutated, and maybe that'll give us some insight into what's causing this in the typical cases. Okay, so one of the mutations that have been studied in most extensively, at least that I know of, is the Volga-German family mutation. So make a little segue into European history. I'll promise it'll be brief. Catherine the Great invited people from Western Europe in the 18th century to come settle in areas in Russia to get the population up, and they could farm and civilize their region to some extent. And one group of takers were a group of Germans who settled in this part of Russia, kind of here near Kazakhstan. They were allowed to maintain their customs and language and religion while living within Russia. So they were kind of an insulated community to some extent. Now, a lot of them, though, eventually left Russia, especially around the turn of the century when there's a lot of immigration to the United States from Europe. And these guys, I think maybe they felt some of their rights were being lost at this point. And a lot of them immigrated to the U.S., particularly settling in the Midwest. So there's a lot of descendants of these Volga Germans living in Colorado and Kansas and Montana and places like that. Okay, so among these descendants, there is a family line with a genetic early onset AD, this Volga German mutation. And it has, by studying these pedigrees with large numbers of members of these families, it's been traced to a mutation on chromosome 1. And then once they found what that mutation was, they were able to find out, well, what protein does it code? What does that protein do? And through tons and tons of research, they found that the mutation is in a protein, a gene that codes for a protein that processes the amyloid precursor protein. And it results in a beta amyloid that's very likely to aggregate. It's a very aggregating version of beta amyloid. So these folks who have this mutation, and it's an autosomal dominant mutation, these folks who have that mutation tend to have more of these plaques. They come earlier. They start coming in their 50s. And by their 60s, they can have very severe end-stage Alzheimer's disease. Okay, so studying this family has really supported this idea that really the beta amyloid is the problem in AD, or one of the primary problems at least. Okay, so those, as I said, that's only a tiny fraction of people who have AD have some big genetic risk. So if you're not a Volga German, but yet you're still, I would say everyone is at risk for AD, given it is so common if you're lucky to live long enough. However, another genetic risk factor is your ApoE, is having ApoE4. Now, this isn't like the Volga German mutation, because for that, if you have the mutation, you will get early stage Alzheimer's disease. This is more like a risk factor. It increases your risk of getting it. Doesn't mean you'll get it, but like the other kind of mutation, but it means you have a risk factor. And this isn't really a mutation. It is an allele that's actually pretty common in the population. So ApoE stands for apolipoprotein E. Everybody has apolipoprotein E, and it's involved in metabolism and transport of lipoproteins. It does a bunch of things in the cell. We don't know exactly everything it does, but it does seem to be important. And it's on chromosome 19, the gene that codes for it. And if I was able to sample all of your genetic material, I would probably find some of you have these three common alleles would be represented. So some of you would have 2 copies of ApoE2. I think most of you would have 2 copies of ApoE3. You might have 1, 2, and 1, 3. Some of you would have ApoE4 as one of your alleles. You could be heterozygous for that or have an ApoE4 and ApoE3. So these are occurring just generally in the population. They're not really a mutation, just like some of us have blue eyes, some have brown eyes. They're just the normal variation. But it's been found that people who have ApoE4, either one allele or even worse, two alleles of that, if you're or homozygous for ApoE4, you have a much higher rate of AD. And that can be like 10 times higher than, say, a person who has 2, especially, seems to be protective, or even 3. So the studies vary, but it's really a huge increase in risk. Okay, so but of course there's plenty of people around who have ApoE4 and live to be 90 and never get Alzheimer's disease, so it doesn't mean you'll get it, but it just sort of increases your chances. Just like, smoking certainly is associated with lung cancer, but there are people who smoke and don't get it, and there are people who get lung cancer and don't smoke, just like there are people who have ApoE2 and ApoE3 only and get Alzheimer's disease, but it is a risk factor. Okay. So what's the problem with ApoE4? Why? Why might it have this risk factor? Well, some views are that it may not be as efficient as other forms of the ApoE in catalyzing the breakdown of beta amyloid. So you might not be able to see this, but this is E2, E3, E4, and it's just a substitute, arginine substitute for cysteine here. And it seems that it may not work quite as efficiently. It's not quite as good when you have this substitution here of arginine. And thus, maybe you're more likely to have the aggregates form because you're just not as good at clearing them out. Okay? And E2 seems to maybe be protective. This is the most common one, is ApoE3. But maybe having the 2 cysteines, you have a really good form of apolipoprotein E. Okay. Environmental risk factors. A huge one, well, the main one is age. Certainly age is a huge risk factor for Alzheimer's disease. You don't see it in young people. You do see it in older people. But I'd say the second most important one is head injury. History of head injury is associated with a high risk of Alzheimer's disease. And you see this in boxers who basically get head injury all the time, even especially concussion. They get knocked out, and boxers have a very high rate of dementia. They get it earlier. They get it much higher frequency than people matched for other factors that have jobs where they don't get pummeled in the head a lot. So one idea of why this is the case is that when you get head injury, it's associated with expression of amyloid. So you get more amyloid protein being expressed when you get a head injury. So you fall off your bike, you hit your head, that's associated with expression of amyloid. And in boxers who have ApoE4, the risk of Alzheimer's disease is almost certain that they'll get it. It's such a high risk. And it seems that here you have a double whammy. You have more amyloid, because every time you're getting punched in the head, get a concussion, you're getting expression of this protein. Plus, you have an enzyme that's maybe not quite as great at clearing it. So I think the risk is so great that I think anyone who is ApoE4 status, this is not the job for them, or any kind of job where you're going to have head injury, because you're almost certain to get Alzheimer's disease. And I think people have called for genetic testing for people wanting to go into a sport like boxing because of this huge risk. Question?
No, just a comment. I think they found the same thing in NFL players and people
and
others who play football. And I think that the NFL is actually suppressing the studies.
Really? Yeah. I mean, I could see also football would be maybe not quite as bad as boxing, but certainly a lot of head injury. I mean, it doesn't have to be like, a car accident level head injury, but just the constant banging, does seem to have an effect on expression of amyloid. Okay, protective factors. So that's the bad news. But what about protective factors? Well, the good news for all you guys, especially higher levels of education, are associated with lower AD risk. Okay, so that's good news. Here you're worrying about your finals, you're studying hard, but you can also take it to heart that you're actually benefiting your brain by all this hard work and studying. And you might say, well, of course, education is associated with better jobs. You probably have better health care, better food. You live in places with less pollution. It's so confounded. Well, even studies that seem to try to eliminate those other factors as much as possible and control for those things still find a benefit of education, and suggesting that the benefit of education is independent of all the other good things that come from having more education, like socioeconomic status. Okay, so, well, why is this the case? And it seems like having higher education, maybe stimulating your brain more, has a beneficial effect. But again, we don't know if is it just because having, if you have a good job, you have a lot more stimulation, you're in maybe a more stimulating environment, and that's why you're protected against AD. Well, some insights came from a very large, long-term study of a group of nuns living in Minnesota who, as part of their good works, donated their time to be research subjects to look at the course of Alzheimer's disease. So it was a large group of women that were studied longitudinally. And, of course, when you ever have a large group of elderly people, some fraction of them developed Alzheimer's disease. And interestingly, if you went back and look at essays that these women wrote when they were very young, say when they were entering the convent, when they were 18, 19 years old, they had to write an essay about why they wanted to become a nun. You could take these essays, and some rater who was an English professor could rate these essays in terms of their complexity, in terms of thought, and some essays were much more or better than others who had much more simple kind of sentence structure and simple thought. So when you rated these essays, it was found that those essays written decades earlier correlated with later AD. Those women who wrote the really great essays had less chance of getting Alzheimer's disease than those women who wrote not such good essays. And why is this important? Well, in the convent, certainly the level of stimulation was the same, the health care was the same for all these women, the food that they ate, their daily activities were all very similar. But the idea is those women that came in maybe with a higher intellectual level, as measured by their complexity of their essay, seem to, even 50, 60 years later, have a protection against AD. So maybe with education, you are kind of increasing your intellectual abilities, and no matter what kind of job you get or what kind of stimulation you have in your life, that will protect you way down the road, 50, 60 years from now. At least that's the conclusion we can draw from this. So this is this idea of cognitive reserve, that we all have some level of cognitive reserve, and that knowing that the Alzheimer's disease pathology develops gradually, that it isn't just suddenly overnight you're full of plaques, happens gradually. There's a point at which the function, the pathology overwhelms neural functions. So if you, at some point you have so much pathology, you're going to start showing signs. Those with higher education, higher levels of cognitive processing, this point comes later. You can kind of function in life well enough until, it'll be later until that pathology develops. So some evidence for that is if you look at patients with higher levels of education who have just had end-stage Alzheimer's disease, the patients who have the higher levels of education, so that you have a PhD and a person who has a high school education, they both died of Alzheimer's disease. That person who has the PhD, a lot more beta amyloid than the person who is a high school graduate. So it's kind of like that person with the PhD could hold it off, their functional decline longer. Okay? That's the idea of cognitive reserve. Okay, so last thing I want to talk about are some of the current treatments that we have out there. So right now, what's approved are treatments that basically target symptoms. They attempt to boost the cholinergic system by inhibiting the acetylcholinesterase enzyme. So you probably know acetylcholinesterase is an enzyme that's in the synaptic cleft, and it chews up acetylcholine. Should be familiar. There are drugs that inhibit that enzyme, thus making more acetylcholine available at the synapse. And that's the kind of standard of care that you would get now. Everyone gets those kind of drugs. However, they're not treating the cause of Alzheimer's disease. They're just sort of trying to boost up the little bit of you have and just slow the functional decline, but they don't actually treat the disease. Newer approaches look into interfering with the formation of amyloid plaques and tangles or their toxicity. And so that's really the focus of the Golda article. What are these treatments that are in various stages of development, either in animal models or in FDA initial clinical trials, to actually get at the cause of AD and try to halt it? Okay, so a lot of these treatments are targeting the beta amyloid toxicity, and there's a kind of a complex figure in Golda. This one, I think, is a little clearer. It's a little blurry here, but maybe you can still see it. Basic idea, let's affect the processing of the amyloid precursor protein. When it gets cleaved up, let's try to cleave it in a way that the resulting fragments don't aggregate. And there's a couple of ways these secretases that are enzymes that cleave it up, there are a few different kinds. There are beta secretase, and gamma secretase, and alpha secretase. And they all cut up the amyloid precursor protein. And when you have these components, these A-beta plaques, they aggregate. So you want to minimize the number of these. So what you want to do is beta secretase, I don't know if you can see it, cleaves right here, and gamma secretase cleaves right there. What you'd like to do is maybe inhibit the activity of these so you don't get these fragments, and actually have this alpha, which cuts it here, because then you won't get that same cleaving. So to try to manipulate these secretases is one treatment that is a huge amount of research is going into that. How do we do that? Because that could potentially stop these plaques from forming. Now, another approach is actually using the immune system, either somehow generating by your immune system to actually attack the amyloid plaques, actually use your own immune system, or maybe a passive kind of immunity, actually inject people with some kind of immune booster that would attack this, so actually using the immune system. Or some kind of compounds that could prevent or break down aggregates. So yeah, allow these fragments to form, but somehow use compounds that will break them up. Okay, so those are kind of the treatment approaches that are really being studied in a lot of detail now. Also, there are ways to target tau. So there is a view that, well, maybe the A-beta fragments that are making these amyloid plaques, that is actually causing the tangles, and the tangles are really the problem. So it's not that these things, it's not the A-beta directly that's causing the cell death. It's going through the tangles, and so actually maybe it might be easier to manipulate the tangles and let the plaques form. Who cares? We really want to target the tangles to save the cells. So reducing the aggregates of tau. Of course, we don't want to eliminate tau because you need microtubules, but maybe we can stop it from aggregating. And there are approaches that are trying to reduce the phosphorylation of tau. Okay, so to stop it from forming these little hyperphosphorylated tangles here. Also, certainly immune approaches, antibodies to tangles is one thing. And another thing is, well, maybe agents that will help stabilize the microtubules. Tau's job is to form microtubules. And maybe there's some problem with the keeping these microtubules stable, and that's causing these little tau fragments to go around, and then they'll just sort of naturally come into tangles. Maybe it's better if we can just make sure that microtubules are stabilized. That will actually help the process a lot, or prevent this process from happening. Okay, so those are kind of the treatment approaches that were described in the Golda article that are, I would say, happening now. They're not treatments you would get in the neurologist's office because they're just still undergoing FDA trials, but actually, when I had been giving this lecture years ago, I could only say, yeah, we don't really have any treatment. It's just looking really bad. But now I think we have reason for optimism, given the development of these transgenic mice that overexpress things like tau and APP. We can really do a lot more treatments than we could before. So this is a great example of how animal models have really helped human health with the development of these transgenic mice. Okay, so the very last thing I want to talk about is identifying preclinical AD. And why is this important? Well, by the time you start to show signs of AD, you already have substantial amounts of cell death. And even if we do find these treatments where we can halt the disease process, there would be a loss of function. So optimally, we would, before the person really is showing real impairment and is really impaired, we would want to give them, so you start these treatments. So an important focus of research in addition is to identify people who are going to get AD very shortly, so preclinical AD. It's been a big focus. There is a group called mild cognitive impairment. This is kind of a new category. So these are older people who are starting to experience a memory decline. They notice something is a little wrong, and their performance is somewhat below normal of what you'd expect for the normal kind of age-related memory decline. But it isn't really affecting their daily activities, and they may not have additional cognitive problems. It can be fairly specific to a memory problem, but they just kind of come in and they notice, my memory, or their wife notices, my memory isn't as good as it used to be. I think there's a little something wrong. So one other kind of hot thing that has been described in the Golda article is the development of what's called Pittsburgh compound B, which when you they call it PIB, but it stands for Pittsburgh compound B, it allows one to detect amyloid plaques in PET imaging. So this Pittsburgh compound B binds to amyloid plaques. So you could take a person who's healthy, who doesn't have Alzheimer's disease. They might have a little concern that their memory is fading faster than they think it should. And combining those behavioral data with this neuroimaging data, you could actually see their risk for plaques. And it seems that people who maybe don't have a diagnosis of Alzheimer's disease yet, but have plaques in particular regions like medial temporal lobe, entorhinal, and hippocampus, that they have a very high rate of soon in the next year or so converting to really a diagnosis of Alzheimer's disease. So using things like PIB will allow us maybe to start, when we have these treatments in hand, start treatments before you really see substantial cognitive changes. Yeah, question?
How early was that predicted? Like how?
Well, the studies now are people who come into the clinic with this, what's called the mild cognitive impairment. They just notice the memory problems, and that's the group that people have been focusing on. You could see in the future, certainly doing a PET study, PET scan has some risk. It has some radiation. So you probably wouldn't want to do this for everyone, but I could see how you might want to do it for people that are a little worried that maybe something is going on. And at this point, we don't have good treatments for Alzheimer's disease. However, we're hoping that soon, some few years, that we will have treatments that will maybe break up amyloid plaques. And so people who come in with a little bit of, starting to have some cognitive impairment could be identified as like, some might be saying, look, you're too worried. You actually don't have any plaques. You're fine. Others would be like, yeah, I'm a little worried. I see a lot of plaques in your medial temporal lobe. I want to get you on this particular treatment. So that's kind of the idea of the future, that this would be a nice diagnostic tool for someone who looks like they're getting close to AD. Okay. Any other questions about Alzheimer's disease? Oh, the files, the help Excel. Oh, okay. Yeah, I just want to make another announcement. Jared has put on the course website some help files for Excel, so running t-tests and making graphs. Hopefully you all feel very comfortable doing that, because you will probably be asked to do that on the take-home exam that you will get in 10th week. Okay, I will have a happy Thanksgiving. Oh.